prions. They're kind of entertaining. Okay, viruses, very small infectious agents. They replicate by invading cells. Since they cannot replicate on their own, they don't really have any cellular structures, not considered alive. They consist of nucleic acids surrounded by some kind of protein coat to protect them. So a virus can have DNA, it can have RNA, it can be single-stranded or it can be double-stranded. So if you see DS or SS, so it can be DS DNA, DS RNA, basic, simple. They have those four possibilities. One other little quirk is if they're single-stranded, they can be considered positive or negative, depending on which strand they do. Some, I mean, it works great because some of those you can use viruses for, we want single-strand DNA for a lab experiment. Well, they create a whole lot of it, so just take it from them. But in general, viruses, they can be DNA or RNA. They can be extracellular or intracellular. So, is the virus particle or the viron, or do they have the genome is released into the cell? So it depends on where do they kind of lose the code.
Do most viruses have some type of injection apparatus to get the DNA or RNA strand into the cell?
Depends.
That's what I know.
Because some of them,
I'm trying to understand.
well, we'll talk more about the different ones. Where do I have them? They're on there somewhere. Because some of them will actually some of them will just stay on the outside and just inject their information in, and then they're done. Others will kind of melt into the cytoplasm, and so their protein structure kind of just melts into and becomes part of the cell's cytoplasm, releasing everything inside. So there's multiple actions for getting their information inside. Okay. Viruses are small. They're very small. I mean, we're talking nanometers. So the smallest have a diameter of about 10 nanometers, whereas the largest are approximately 400 nanometers. And if you think about this as about this, so their largest is about the size of the smallest bacterial cell. So overall, you're like, okay, really small. Again, their genetic information is either single or double-stranded, DNA or RNA. We used to only think it was DNA. Finding those RNA ones has been kind of entertaining. They're called retroviruses. And usually with the RNA, if they have RNA as their genome, they're also going to come with an enzyme called reverse transcriptase. Because what is the central dogma?
DNA, RNA.
RNA to protein. Okay? RNA viruses go against that central dogma because they're RNA. And they have reverse transcriptase, which does like what it says, is reverse transcription. It goes from RNA to DNA. And then once they're in the DNA format, they're able to then integrate the genome and stay in there for a while and then come back out and do DNA to RNA proteins. But that reverse transcriptase has been very useful in the scientific community, but also one of the reasons why RNA viruses are such a pain in the butt is that reverse transcriptase doesn't have a proofreading function. It makes a lot of mistakes going from RNA to DNA. So within one patient who has HIV, they can have, I think it's like hundreds of different versions of HIV just because of all the mistakes it makes as it's trying to do that transcription. Okay? So, again, we're just kind of looking at the sizes and the difference between our red blood cells. We're talking about 10,000 nanometers in diameter versus E. coli, which is, 3,000 30,000 nanometers. What is that in micrometers?
30,000.
So nano is 10 to the
3,000 would
9.
be 3.
Micro is 10 to the negative 6, so you remove
30,000.
3.
3.
Remember, math is on your test. Okay, so they're between 1 and 3 micrometers, but we're talking about nanometers in this case just because we're looking at viruses, which are really small. So you got tobacco mosaic virus, you know, might be as long. Nope, still not. It's a third the size this is a crossed. So smallpox, they're incredibly small, but you can see some of the different ones and how they'd actually function. Bacterial ribosomes can be pretty small, so they're on the same size as an internal component of bacteria. But
Like a polio.
Hmm? yeah, polio is very small. Bacteriophages. Bacteriophages can infect, they're viruses that infect bacteria. T4 phage is common. But you can see all the different structures, from nice symmetrical to a head and tail sections to very weird long ones. We'll talk about more. So, structure. We have a capsid. So we have this protein coat surrounding the nucleic acid. You need something to protect the nucleic acid. You don't just want DNA or RNA floating around free because it's not the most stable. DNA is much more stable than RNA, but still you don't really want that out in the environment because it'll degrade. DNA you can leave on your benchtop and it'll probably be fine for a while. RNA you look at funny, and we have a lot of RNases in our bodies, so those would be degraded. If you have the nucleic acid plus the capsid, it's called a nucleocapsid. And then we have envelopes. A lot of times that will cover this. So you have your capsid, which has your information inside, and then you have envelopes around it. So that way it's even more protected. So these envelopes, they differ from virus to virus, what they have, how they have it. But in general, this is why you can have an enveloped or a non-enveloped virus, because they all have that little capsid to help protect them, the capsid. So capsid, the entire coat to protect your nucleic acids, but then some of them also will have an envelope to help them along. So when we were talking way back when about the difference between an enveloped and non-enveloped virus, they have that extra layer or not. So this was, I like this because it showed just some different types of viruses. Tobacco leaf virus, the mosaic virus, tobacco mosaic virus. It was the first virus discovered because, well, kind of like our tobacco. It's useful not just as a tobacco product. A lot of biotech and research is done in tobacco because they have these nice big leaves. They make millions of progeny by seeds, and so they're great for genetic studies. But having a virus that kind of chews up leaves and makes it so that it's not very productive is a problem. So there's been a lot of study been done on the tobacco mosaic virus. So this was a cute little picture of bacteria cell. It has bacteriophages. That's not as fun. It's a ton of little bacteriophages, and you can see that they just all kind of come around and start shooting their information inside of the cell. Versus if we have white blood cells, so a human white blood cell, and it has a bunch of HIV attached to it. So actually, it's only been in the last 5 or so years we've actually gotten visualization of HIV attaching to cells. So some of that's even, it's still pretty new and novel to be actually able to see the attachment to the cells and try and figure out how can we stop that then. So, different viruses, they affect differently. Virus structure can be very different. You can have helical structure for your tobacco mosaic virus. Use it all down. Polyhedral. Buckyballs. So the adenovirus common cold. So you're getting, so just very geometrical shapes. Or you can get very complex one, like a bacterial phage, where you end up with, a head, and tail, and then tail fibers, and you get